partner, techno provider. Thank you, Thierry, for joining. And,
okay, thanks a lot for your welcome. So I'm happy and thank you for giving me the opportunity to present Cilios and its technology. So in the Invite project, in fact, we are a techno provider of GVS and INRAE for the Fusarios. But I will disclose that a bit later on. So Cilios, I will start quickly. So by who we are just a small company, born 2001. We are in the area of Aix-en-Provence, which is in the south of France. But our core business is micro-optics. And micro-optics to do what? To do first part, the diffractive optical element for laser beam shaping. We are part on face plates. We're in the, let's say, very high and intense power laser worldwide, and also in the astronomy field with the extremely large telescope, VLT, etc., to do some faceplates. But also the other line of product is the multispectral imaging, so with a techno that we have developed, which is called color shade. So this will be the topic, and this is what we have applied within your Invite project. So I will go quick on that. So what we can supply, in fact, we can supply custom and off-the-shelf products, so custom like for this Invite project, but also off-the-shelf products. So in fact, our business is to do this small piece of glass and align it pixel filter to pixel sensor. So at the end, it does something like that. So quite tiny, quite tiny. And we can supply also multispectral board, which are in fact a complete camera without its enclosure, so that we supply to people who have some type of drones, so that they do their own enclosure, IP68, etc., and the connection to the PC board of the drone. And we can also supply multispectral camera. So these are the wavelength range that we address. We have a first type, so with we call it CMS and CMS4, the same spectral range, 8 spectral band, with 1.3 megapixel and 4.2 megapixels. The other wavelength, which is more the interest of the people that work in agrofield and agronomy, 8 spectral band from 500 to 800, in which you can have the NDVI and other index that are useful for your application. Another one from the 600 to 900, 930 exactly. And finally, really brand new sensor that which is currently unique on the market in the type of, in fact, a snapshot multispectral biometric type is the Toucan. We call it the Toucan. It's 10 spectral band from 400 to 900. It's the only one currently on the market, available on the market. So to give you an idea of size, this is the one of the CMS. This is the size of the CMS, quite tiny. Easy also, something few grams to be handled on the drones. And this is the Toucan, the size of the Toucan, the 4 megapixels. And also the weight, something like less than 170 grams for the one. So I will present you some application we have made in the smart agriculture and also agro-food. So what I spoke to you early, so the e-boards for, let's say, plant health monitoring, biomass, let's say, the start of the precision farming. An application of the CMS4V, so the range, the band range is 550, 830 nanometer. One of our customers, so we used to work with the RGB to detect, in fact, early disease detection in orchards. So it's mainly proximity detection. And they worked with the RGB, so it's not so obvious to find the disease. And just with the false color, without any processing, you see with the use of the other spectral range that the disease, really, there is a nice contrast. And this is very important, because if the eyes sees it like that, that means that when you pass it to your AR, let's say, artificial intelligence process, there is a better discrimination. So this is, in fact, it's a project that we made with Wageningen. So here when they apply their process, so they find very easily the disease. Here, the big advantage also is that under light or within shadow, you still have the same discrimination. Which is more difficult with just RGB. Another application is the let's say, early disease detection also in vineyard. So here what they are looking for, in fact, is the flavescence dorée, which is a big problem within Europe, mainly, and also other diseases like escale. So again, with their process, the customer discriminates even, let's say, the disease in a plant, and in a leaf, sorry, whether in a dead leaf or a live leaf, you don't see the difference. So that means that when it's not infected, I have to go quick.
No,
okay,
you have to go.
I have to go. Okay, so and then he improved the system. It's bad because it's the one from Chives. Okay, allez, sorry.
No, it's to highlight. So you can of course join Thierry then in the room.
So that's what.
More discussion.
Yes.
There will be no question. It's just to highlight what will be the content.
Okay.
This kind of.
Well, anyway, everything is over there. You can see it, except this one. You will not see it, but just ask to Chives.
Thank you.
You're welcome.
So no question now because there is 2 hours and we want for discussion. That's
yes.
Thank you. So this kind of camera, please, well, simply not accessible at affordable price. Just 5 years ago. So that's really super nice to see that it optimized multispectral camera now can be done for your own purpose, which is completely recent technique. So we stay.
Actually, I think
we now back inside an insider of the project
this
with
presentation.
Mourad.
I'm searching the mouse.
I'm not trying.
Yeah, here.
Okay.
T'as mis la présentation, Thierry Moi, techshow. Ouais. Oui, oui, t'inquiète, j'irai rapidement. Can you make it on full? So, hello everyone. For those that don't know me, my name is Zinel Abidin Mourad. I am a PhD student currently at my 3rd year, almost 5 months to defend my thesis. So my contributions are included in the work package 2 with deliverables including annotated data and trained models. But besides this, I also developed 2 applications, Panovar and Ordinalysis, to answer the need of a variety testing community when we want to deploy machine learning in such applications. So let me, before going into details, I will quickly just remind why we do all this. So this is me with some bad acting, just trying to prove that we have a complexity in variety testing regarding the subjectivity, and it's labor intensive, and so all the other factors that you know. And we want to exploit computer vision to make it fast, objective, and especially reproducible, as we can reproduce the same phenotyping, digital phenotyping tests all over the Europe sites. So regarding computer vision, we exploit what we call machine learning, which is basically some codes that are able to learn over examples. And what is more robust is supervised machine learning. And supervised machine learning means that we have, for instance, images, and we have the associated classes for region of interest. For instance, we have some pathology progression, and we have class 0, class 1, class 2, and so on. So this is the experts that do this operation. And the problem is during the machine learning deployment, this annotation is not, or the comparison is not fair enough, because when we apply machine learning, we apply it on the images. But when the experts do the annotations, they do it by visual inspection. So somehow the comparison of the results of machine learning is not fair. So this is why I had the idea of developing Panovar, which is a web application dedicated for variety testing community, where the experts and the users can annotate or do the measurement of variety testing. Besides this feature, the Panovar also allows the user to quantify the variability between the annotators. For instance, we have now, just for example, GVS and INRA, two experts, and we want also to quantify this variability between two annotators so that when we compare to machine learning, we know if it's machine learning is doing well or not. And just to finish with the feature, I think it's the main one, is to implement machine learning also to offer the possibility to either annotate by the expert or also launch a machine learning model that was trained on previously on annotated data and also was verified. I will move quickly to the second application. Oh, yeah, sorry, this is just screenshot. You can see this more details in the techno showroom. But this is just the demonstration of this web application. Panovar was on the apple shape characterization and with collaboration with the IRTA, represented by Maria Jose, who already experienced this application. So as you can see here, the user have the option to load images and choose the right classes. These classes, the drawings that you see, are just cropped from the catalog of variety testing. So it's somehow we mimic the same procedure. And to finish with the second application, this second application is more dedicated for users that want to know what is happening in machine learning. This is why we propose Ordinalysis. Ordinalysis offers the possibility to load images and understand the structure of the feature, what we call feature space, visualized feature space. If I can just quickly explain, this concerns the ordinal data. You know, in pathology tests, we have ordinal scale, for instance, resistance, moderate, severe. And visually, we already know the ordinality because we see progressive progression of the area of diseases. And well, hopefully for us, we also expect to see some progression in the feature space. So as you can see some clusters here. So we already know what we expect somehow, what the ordinality should look like in the feature space. This is why we take benefit from this expectation to compare different machine learning techniques to choose the right one, which is, of course, producing the best representation of ordinality. So I will provide you with more details in the techno show. Thank you.
Hello everyone, my name is Adami Garbouj. I am PhD student in the University of Angers, and with my supervisor, Dr. David Rousseau. Okay, okay. So in this presentation, I will present 8 tools dedicated to the tasks done in the growth chamber. So in the beginning, I just try to introduce our context. So the current technique in growth in this control environment, it's do a lot of measurement manually. So as growth measurement, development stage, and disease detection, and all of this need to biologists to go each time to the ground truth. So it's very time consuming. Donc, in our task is try to automate this several tasks. So for that, we implement several RGB infrared depth sensors in growth chamber, and related to mini computer to manage or acquisition and to also manage camera remotely. And we saved RGB infrared depth image each 15 minutes, and we can change all this time lapse. And we send it each hour all this image to the server to be shared with biologists. And also to be sure we have all of the day images, we check our camera at 2 hours, and if we have any problem, we received email to, you should just see why this camera don't works well, and also we receive each day email from your server to check the situation of each camera. And after that, we do several machine learning models to dedicate, for example, the stage of growth, but we cannot send it to biologists, just this code or this model in Python. So for that, we built a software to facilitate the utilization of machine learning tools with biologists. So just one information, the cost of this room, I think in this room we have 8 cameras with 8 mini computer. It costs from 500 euros. So this is our software. So with this software, we can, for example, detect automatically the trace in our room, and also the individual cells of germination. And also we can have the link with each cells and each genotype he was on this trace, because we can implement also your plan, experience and plan in this application. Also, we can have the growth curve of your plant each day, and also if you have during of 2 weeks. Plus, we can also have the stage of development each day and each time when we have acquired images. And other several features can be also added in this application. So thank you for this moment. And in the room in our show, techno showroom, I will present the sensors, with very small sensors, and we use it in the field and also in indoor condition, and also the software. Thank you.
So we continue on the same kind of sensor, which again was existing 10 years ago, but in a bigger, larger size, and the price continued to decrease. And somehow all this software will be available in a couple of months, so accessible. They were sold by bigger companies earlier, but now this kind of open source spirit becomes accessible for everyone. So with the same kind of sensor, we move to Vur with Joseph. Thank you.
Yep. Hi, everyone. So as he mentioned, I'm working on work package 2, and our part of this work package has mainly been looking at tomatoes and working with Nachtenbau in the Netherlands. And when we're primarily looking at post-harvest. So as you can see here, post-harvest analysis of traits normally requires a very labor-intensive time. You have a few examiners who walk box after box after box, score the traits one after another, and they're experts, it's what they're paid for. But there are also traits that are easy to score that takes no time at all. And then there are traits that are very difficult and takes a very long time. So before we started this process, we kind of talked to the examiners. We figured out what are the things that we should and can't, what would be good to basically tool on. And from that, we've developed this tool that I'm going to present in that room over there after this presentation. So basically, our tool is going to focus on these traits that I've listed on the right side. Those are the ones that are either hard or take a long time for the examiner to look at, with the exception of color, which we recently decided was really easy. So what we have over there right now is a prototype. It is simply a RGB camera that has a depth sensor into it that is connected to our computer, and it's running a piece of software. Our goal, though, is to actually implement this on a mobile device. So we're either going to put it on a phone, which would be ideal, and there are new phones out there that have 3D information, or to put it on some kind of tablet. The tablet was initially an idea because that's what they're currently using in Knox, so it's an easy transition towards new tooling. What our actual program does, though, is it scans a box of these tomatoes, and then it segments out each of the tomatoes in the box automatically using machine learning. Then it can detect whether or not those tomatoes are upright, on their side, upside down, basically what is the orientation of the tomato over the entire batch. From there, we can break it apart into 2 traits that we're going to show you over on the other side. One of them is peduncle size. So we run a separate network that looks at any upright tomato and says, here's the peduncle in it, measures it, and because we have a depth camera, it can give us an actual like millimeter size of that peduncle. And then we can use that to recommend a score to the examiner. The other thing it does is volume and shape ratios. Tomatoes' volume is directly tied to its weight because the density of a tomato is 0.99999. So like, that's its weight. But then also the shape ratios, because tomatoes come in really funky shapes, and knowing the correct like shape is a very important phenographic trait. So to do that, we basically take the side tomatoes and the upright ones. We can draw these two ellipses on it and basically figure out which of these ellipses matches the one on the other side. Throw that one away, and you have a 3 ellipse and kind of can remake an ellipsoid of your tomato. And from that, we'll then match it back to the traits that are described in UPOV, and we have yet another trait that we can recommend. And again, the goal is to put this all in a snazzy app that you can then just give off, and then it will just, you take a picture and you get a list of recommendations for each of the traits that we have. That's what we're showing in the other room. I'm done with my presentation.
What is yours?
Then it will be hyphen, and then or Benoit, I don't know, but so you need to come with the stick, I guess, to use it.
David, that is the name. Just not this one, slideshow, please.
I don't know, peut-être. Et sinon, il faut que tu le remettes. Ou je sais pas, où je l'ai peut-être coupé. Tu l'as sur une clé ou ouais.
Mais dans l'autre.
Ouais. Ah, bon, d'accord. On change tous les deux si tu l'as sur la clé. Ok. Sorry, Philippe. Ok, vas-y.
It's here.
Which was already, let's say,
That one?
preparation when we started
Ah, yes,
the
the first
project,
one.
that was not included as a tool we would develop in our package 2, but which is now ready, so we wanted to highlight it
Okay.
to you. And so thank you,
If you can
Benoit, for
presenting
presenting this new tool.
I think it's okay. It's okay.
Hello, so this time I will not disconnect the screen for the one who were in the mice session this morning. So welcome everybody, and thank you for your invitation to present this tool. So Literal has been developed, so I'm working for Arvalis, at Arvalis, and we developed first this tool for our own activities. So we wanted to replace visual assessments in the field that are done by the technical staff using, yes, an affordable and easy to use device. And that was yes. So that was the aim of this development. So to have an affordable, simple, and reliable phenotyping tool adapted to small or medium-sized trials that is not covered by UAVs or by heavy phenotyping systems. So something that is also able to perform some measurements in farmers' fields in case of a network. And therefore, we designed this system to integrate high-resolution images, to facilitate the use of deep learning methods and power supply, geolocalization, something light and flexible to adapt to different crop heights or configuration. And then we start developing data processing chain to derive interesting traits. So this is the system that we are now using in our field trial. So we have deployed 27 systems like that in Arvalis network, plus about 25 other systems in our partners' sites. So yes, roughly 50 systems are now working. So it's basically a stereo camera with some also GPS, accelerometer inside that allows a good referencement of data. And then we have a tablet that control acquisition to select in the field trial map which field and which plot is concerned by measurements. So we can then decide for each microplot which is the measurement scenario, and with that we can really control that each microplot has been phenotyped in the same way. Then we developed some data processing modules. So these are the basic ones. So from high resolution images, we were able to develop some detection modules using deep learning, also segmentation, also using deep learning, and some stereo vision modules to characterize the 3D architecture of the crop. Okay, then here are some examples of applications. So wheat heads counting was one of the first one because it was the most demanding by our technical teams. Also flax plant counting. So these are now used. So crop cover fraction estimation. So this is full vegetation estimation. Then we have another module that discriminate which is green and which is non-green to monitor the senescence. And then we have all kinds of 3D analysis based on the stereo vision module. So to estimate the crop height, but also to go more in detail on the architecture of the crop. And then we have developed many more modules. For instance, here a module to estimate the leaf senescence. So we have a module which segmentate separately ears, stems, and leaves, and from these we can estimate really the senescence of the leaf part. Here it's a module to segmentate only the maize crops to discard the weeds and really estimate the leaf area index of the crop itself. And here we start to develop applications for mixed crop monitoring, which is a growing concern in our farming systems. So to estimate independently the leaf area index of each crop. So thank you, and you are welcome for more information on the device.
Maybe here? USB? No. We don't have it. No, the USB key doesn't work on this. I don't know why. Maybe. Could I try? The other way. No. Yeah.
Where is it? Here, that one? No.
Yes.
RGB. So much.
So to move is
This is here.
yes. Okay, sorry for that. So my name is Philippe Vermeulen from the Walloon Agricultural Research Center in Gembloux in Belgium. I am the task leader, the task 2.4 in the work package 2. And the idea of this techno-show stand is to show you the different applications we developed and test during the 2 first years of the projects. So there you have a list of different devices. So we worked only on non-RGB devices, and we test those different devices to assess different parameters for different species. So you have the height for white gas, the number of apples, and the quality for apple, and also the fusarium detection for wheat. And so I would like to thank you to the people from INRAE, from GEVES, from Microscope, from CREA, and also my colleagues in CRAW to collect all those data and to develop the methods. So for each method, we write a protocol summarizing the way to acquire the data and the way to process also the data. And so for each example, you have such protocol. So we have a protocol dedicated to the height of the wiregrass using photogrammetry. Another one dedicated to the numbering of apples using LIDAR technology. And another one using visible near-infrared spectrometer to assess different quality parameters, for example, the dry matter and the bricks on apple. So regarding the study on the fusarium, we tested the different devices. So you have 2 first devices. It's point-to-point measurement. So we use handheld instruments to make the measurement directly on the ears of wheat. Then we tested the different imaging system in the laboratory to measure the stress on the ears, so the fusarios in this case. And finally, we test also the same imaging system in the field in order to have a different view. So you're using different vectors, and you can have a frontal view, a perspective view, and a vertical view. So in particular, during this techno show, you will be able to learn more with Frédéric Boudon to assess the number of apples on trees, with myself to know how to assess the quality of apples, and with Valérie Cadeau and myself to know more about the assessment of fusarium on wheat. So you are welcome in a few minutes in this room. So the AE 58. Thank you.
Do you see it somewhere? Yes? Yeah.
Thank you. Hello, everyone, and thank you for the invitation. Today I want to present you very quickly the different digital phenotyping tools that we offer at Hyphen for variety testing. So my name is Alexandra Burgi. I joined Hyphen, a French-based company, 1 year ago as an R&D project manager, and today we are a team of about 20, a bit more than 20 EgTech experts, especially IT scientists and agronomists. And maybe you already know Alexis Comar, the founder and CEO of Hyphen. He's not there today, but maybe you know him already. So at Hyphen, we do digital phenotyping by image analysis. So this is to say that we transform agricultural images into valuable phenotypic data that you can then save time because it is much more quicker than manual phenotyping. You can scale up. You can also improve labor efficiency, and we can deliver results within 3 to 30 days. So we work with all types of sensors and devices, so a lot with drone images, but also with satellite data, with stationary field systems, field robots, or also more and more with images that were acquired from smartphones or other handheld devices. So we already offer a wide range of different traits. For example, the green vegetation cover, the plant height, the bio volume. Also, we can count plants or plant organs such as weed heads. And also really interesting is the so-called agroclimatic traits. So here we combine the biophysical traits with climate and soil data. And with this, well, we can better explain the plant's development because some factors that are linked to climate or soil can be eliminated. Once the traits are calculated, you can find them all in one site in Cloverfield. This is our web interface, where you can visualize the fields, the different traits, and also download the results. And soon we will also integrate some statistical analysis, for example, ANOVA, that you can get really early in the breeding cycle, a quick idea of your trials. But this is not all. When you start working with us, we will always advise you which sensor, which device will best suit your needs. And then, for example, if it's for you the first time that you use drones for plant phenotyping, we will explain you or show you step by step how to implement drones in your research program. And also, you will have free access to the Hyphen Academy. This is the first e-learning platform to learn how to acquire research-grade images for agriculture. So today we offer 4 different catalogs. So one, the PhenoScale catalog, it's about using drones for agriculture. Then PhenoMobile, so these are field robots, and with these type of systems, we get high-resolution images, so we can, characterize plants in a more advanced way. Then we offer also phenostations, so this might be interesting for vertical breeding. And then if you have some more ideas, maybe you want to develop a smartphone app or some new traits, we can also help you thanks to our Pheno Research catalog. So don't hesitate and come speak to me just over there in room AE56. Thank you.
Session. Should have it. Yeah. It's okay.
Okay. So, hello everyone, and thank you for the opportunity to speak here. I'm Iji from the company Photon Systems Instruments. For nearly 30 years, we specialize in development and manufacturing of the instrumentation for plant and algae biotechnology for different scales, from single organelles to whole population of trees. The company was founded in 1994 by current CEO. We are located in Drasov, probably 2 hours from here. We were the first company to offer pulse amplitude modulated chlorophyll fluorescence imaging. PSI currently has around 100 employees in all fields of expertise, and we also run our own research center. So we can utilize devices we make. You can send students to do the experiments if you don't own the phenotyping system, for example. So For the introduction, I have selected 2 installations we have. These are pictures from Australia, and this is the picture from Expo in Dubai. And the first installation I will talk about in detail is one located just 2 or 3 speed traps from here. It is in Vienna Biocenter core facilities. This is full controlled environment phenotyping system with basically all sensors: RGB, chlorophyll fluorescence, visible near infrared, shortwave infrared, hyperspectral imaging, and also extensive options of watering and waiting. So this is the basic layout where we have the growth area with lights and the phenotyping part with all the imaging sensors, and plants cycle automatically in this controlled environment. This is the process of the installations. This is where we made the climate chamber and the illumination. We put the conveyor systems there, and now it's operational and running. The next installation I have selected is in IPK Gattersleben. This is a different approach. Here we move with the imaging sensors to the plants, which are sitting in a big containers. We also made special hybrid illumination there, which consists of metal halide lamps and a multispectral LED illumination. This is, again, from the construction. You can see the illumination and the phenotyping crane carrying all the imaging sensors. And this is the first experimental trial. As you can see here, there is also shading, so we can do side view imaging, which is quite extraordinary with this layout. We are also partners in several European and international projects. One of them is ADAPT or Capitalize. For now, that's it. I'd like to invite you to see us here, where we present few of our smaller devices, and you can also send me any questions on this email. Thank you very much.
There will be a stand of the PSI company inside the room. So we move to Pixofarm. He develops special tools for Apple. When you working with the company and occasion to see, let's say, tools, much lighter tools adapted to a specific trait of interesting device. Thank you.
Good afternoon. Thank you for the invite. Great name, by the way. My name is Georg Kanner. I work at Pixofarm. Pixofarm is an HTech company, a startup based here in Vienna. We specialize in yield monitoring and estimation. Through our smartphone app, we empower fruit producers globally to meet their goals and produce sustainably. With our services, we increase efficiencies along the entire value chain. Pixofarm has been recognized by industry-specific juries like the Interpoma in Italy, you might know, or more general ones like the Global Startup Awards, and we are active in over 20 countries by now. Here are the services that we provide through our app. Users can size fruits with their smartphone or do fruit counting just by taking pictures of their trees. Our algorithms calculate precise yield estimations based on these inputs. We are currently working on our harvest bin scan, which allows us, our users, to take pictures of crates filled with produce, and the system will count and size the top layers that are visible. To get started, users follow 3 quick and easy steps. They create their production blocks in our system, and they're good to go. Start taking pictures in the blocks and get the results immediately. Now, why are growers doing this kind of work? I'm sure you know this. With this information, growers can optimize on-field operations, get a better yield. They can negotiate a better price by referring to the yield estimations before harvest. They can optimize picking and packaging processes and storage procedures. Here you can see also what we mean by working alongside the entire value chain. Here's what sets us apart from others. To highlight just a few, with us there's no need for extra hardware or special trained personnel. There's no limit in the number of people in using the tool or in how many pictures are taken, and you have total control over your data and all the processes, and that which allows you to take more informed decisions. Now, how does it work? For fruit counting, you just line yourself up with a tree and take a picture with your smartphone. The system detects and calculates the fruit count from there. Here are a few pictures showing what the result looks like afterwards. You can later visit me in the hall and I'll show you videos and the thing in action. Fruit sizing can also be done with a smartphone. Here you move a bit closer, move a bit closer to the selected fruit and take a picture. By monitoring fruit sizes and growth rates, growers are optimizing on-field processes again to get the best yield. This can also be done with a Bluetooth caliper, so I assume most of you have been using calipers for now. That this caliper you just connect to the app and then just press one button while the phone is in your pocket and you receive the measurements and you go very quickly. Here again a view of the results of fruit sizing, what it looks like with Pixel Again, later can show you more details on this. And then next up are the yield estimations that we do. By taking into account the ongoing weather data, the growth behavior of a crop, and the measurements that the user takes, our algorithms calculate a reliable yield estimation weeks before the harvest. We predict yield volume, average size, and size case distribution at harvest. As mentioned earlier, we're developing a harvest bin scan, similar to a previous speaker has been doing with tomatoes. This allows users to take pictures from the top of full harvest bins to get fruit sizes and fruit counts from the top layers, which is then calculated for the entire bin. Interest in this service is quite high since sorting is a big bottleneck at harvest. And last but not least, of course, data management. So this is clear. All of the data is easily available, either in the app or on the computer, on the online dashboard. You can view it in any browser from anywhere and share the data with any important stakeholders that you want to share it to, or compare production practices internally as well. Thank you. I will be in hall 38 too. Perfect.
Thank you very much. So we have the last company for the Vienna Center, because I skipped one at the beginning, and then we will have our neighbors as last presentation. So I just have a short video from a company, and then it will be you. Okay. An old guy. Okay, so that's this. So we could not leave without talking a bit about maize. I hope Claude is still with us. Yeah. Where are you? Yeah. And so this is a last small company called Fimea, developing some tools in.
Hi, my name is Sebastian Lecure. I am an associate at Fimea Systems.
How can I put the sound a bit higher? Yeah, sorry. Can you boost it a bit?
I think we have to
Well,
close that.
just a bit. It's just
Louder.
I don't have. Yeah, you're almost there. Yeah, maximum might be good. Thank you. Thank you. Okay.
My name is Sebastian Lecure. I am an associate at Fimea Systems. I'm sorry I could not be available.
Yes?
Yes. All close everything.
Today, but I hope the video can keep you interested. We are a young company aiming at building phenotyping systems for plant science.
Great. Thank you. Okay, so let's
Hi,
go.
my name is Sebastian Lecure. I am an associate at Fimea Systems. I'm sorry I could not be available today, but I hope the video can keep you interested. We are a young company aiming at building phenotyping systems for plant science. We mostly work with open source tools to provide low-cost hardware. We also develop software and image analysis tools. We have been working on various projects, and the one I'm here to talk about is the project we had in collaboration with INRAE Diascope in Montpellier, for which we developed a phenotyping system for high-throughput maize ear phenotyping called the Earbox. So what is the Earbox? As you know, to perform statistical and genetic analysis in general, on maize ear phenotypes also, you need a great deal of data. And phenotyping manually 50,000 maize ear is quite resource consuming, both in terms of money and time. So the Airbox resolved the problem by increasing the throughput and the precision of measurements. It's a dual system composed of an image acquisition station that you can see there, and a software for analysis that allows a higher throughput and more precise measurements than manual estimations. The whole logic of the acquisition system is to acquire normalized images of ears, their whole circumference. For this purpose, they are placed on rollers, and images are taken on 6 sites. If you're familiar with it, the system is composed of 2 Raspberry Pis, imaging 6 ears at a time in both the RGB and the infrared spectrum. And this can be customized for 3, 6, or 9 ears at a time for various throughputs. And in these configurations, the system takes 30 seconds for 6 ears. So if you account for manipulation time, putting them together, scanning their code, and taking them out, you end up with around 10 seconds per ear, so around 6 times faster than manual measurements. So something that would take 1 week will only take a day. And for the software itself, as it demands high computing capacity, the analysis is done afterwards, separately. One must take the hard disk from the Earbox machine and plug it into a computer. From there, we have a software for the analysis to output traits of interest. We have 2 models for clients. Analysis can be performed either on Famia's infrastructure, after sending us the images, or on an analysis station set up in the lab from which you can directly extract the results. Obviously, licensing can be discussed and fixed depending on labs and infrastructure needs. So which variable do we measure with our software? Well, actually a lot. We've built a machine learning and deep learning algorithm trained on a large phenotypic diversity that allow measurements of all typical variables of the ear, length and maximum diameter, and the grains, such as the number of ranks, rows, number of kernels per rank or rows, and number of kernels per ear. We also provide characterizations for abortion, fertile length, and estimations of basal and apical abortion lengths. For people interested in ear and grain colors and types, we can provide measurements of grain and cup colors based on our in-house classification algorithms. As those are specific measurements that can be dependent on client needs, can also be customized with specific classifications. And finally, with image analysis, we can actually go further, much further than this, and output data of grain individual dimensions and their positions along the ear to study grain development. And we've also developed a new variable that takes cob and grain pixels into account to estimate the grain set ratio that can be used to precisely analyze abortion along the ear. And if you're interested, we have a paper on the way that just came to be revised. So I'll invite you to take the link on the QR code on the poster if you want to know more about the methodology we developed. So how precise are we? We tested phenotypic variables against manual measurements for both development datasets on the left that you can see in our paper, and on the right for new data, 1 point per variety, which was acquired last year with GVS. It precisely shows that you can expect great precision for the outputs, which are comparable to what you can get manually, but with higher throughputs. And one advantage also is that you can avoid experimental effects, that you can compare experiments together, as the machine will always apply the same methodology. And we're actually especially happy with the kernel per ear calculation we have here for both the development and the validation datasets. So what's next for us? Well, we have been busy at the end of the year building partnership to improve our system and to provide new traits and new crops. We have an engineer working together with us and David Rousseau from Laris Lab and University of Angers, in collaboration also with CRAW in Belgium, to develop methodology for acquisition and image analysis for wheat, barley, spelt, and other cereals. And we've built a partnership with CRAW in Canada to enhance our maize algorithm and provide recognition and quantification of ear pathogens, such as fusariosis detection, and western bean cutworm estimation of damage. If you want to know more, there's a YouTube video of the yearbook system we just made, and you can also check the poster and come back to us if you have any question. If you want to test the system, we usually provide a possibility to test it at your place by renting it for small experiments. Thanks a lot for listening and for the invitation. I'm available for any question, and I hope you have a pleasant end of day.
Okay.
Hi,
so
my name is Sebastian Lecure. I am an associate at FIME Assistance.
There is a poster that the company sent to us. We're working with them actually at the moment, so I'll be able to answer some of the questions on the work, especially on what we do on wheat at the moment. And we welcome the last speaker of the session from the Vienna Biophenotyping Center. Okay. Where exactly? So here. Do you see your stuff?
Yes, here.
This one?
This one, MCF.
Yep.
Okay, how do I move on? Here?
Yep.
Okay.
Control L. Full screen. Yep.
Yes, so my name is Jacob. Thank you for inviting me. I'm obviously from the Vienna Biocenter. The Vienna Biocenter is a world-class research life science center in the heart of Vienna and in the heart of Europe, hosting different world-class research institutes like the IMP, IMBA, GMI, and Max Perutz labs. Most of them focusing on life sciences research like regeneration biology or cancer research. Organoids, brain organoids are coming from here. And the GMI is the only institute mainly working with plants. As you see, we combine research, education, and also businesses, start-ups or spin-offs. But the slide intends basically to show you that such world-class research needs also high-tech research infrastructure. And that's therefore the Vienna Biocenter core facilities have been implemented 10 years ago. So what we are doing is we are managing, organizing, and further developing the research infrastructure on campus. We are organized in 10 scientific core facilities, from advanced microscopy, electron microscopy, next generation sequencing, up to metabolomics and preclinical phenotyping. And I'm heading actually one of those facilities, and it's the plant sciences facility, and that's why you invited me and not the other heads, I guess. So our main field of expertise and our service portfolio focus is on the environment and on the phenotype. We are most of the time in the field of basic research because the GMI is doing basic research. But as I will show you, our infrastructure is also suitable for crop plants and for other applications. So we operate 23 really high-tech and highly precise walk-in phytotrons and also 7 glasshouse cabins. The phytotron that you see in the middle is one of our specialized phytotrons that will basically introduce soon. It's equipped with high-tech LEDs, with variable LED light. There is a XYZ RGB robot going through the chamber. I will show you a video in our presenting room just next door. And it's also a frost stress chamber, so we can go down there to minus 15 degrees. And that's the environmental simulation service. I mentioned already we have 4 chambers with minus 15 degrees. We have 1 heat stress chamber to heat up to 50 degrees. We can do a lot with the spectrum and with the intensity, and we have also phytotrons where we can control CO2 levels. This can be done in a very precise designed way, experiment designed way, or we can also simulate live environments like we, for example, do regularly for Magnus Nordborg, where we simulate Swedish spring conditions in Austria summertime, for example. So we are also the coordinators of the emphasis national node, and we coordinate the Austrian Plant Phenotyping Network, where AGES is also a partner. Where the most and main of the Austrian research institutions working with plants are partner, and out of the APPN and with the support of all these institutions, we went for an FFG grant of 1.5 million to build a national infrastructure for high throughput plant phenotyping. And this was introduced already by PSI. It was one of their examples, but you know what you hear twice, you remember maybe easier. So I will not skip this. It's a huge phytotron basically because we are familiar and we feel comfortable with highly controlled environments. So we built the system in a highly controlled phytotron and not in a glasshouse. We have this buffer area on the right side where the plants are growing under highly precise controlled conditions and LED light and are transported through the imaging boxes, being analyzed in different means and then transported out. There are 2 watering and weighing stations where the pots are being weighed and watered up to reference values that you set, and then they are moved back to the grow buffer. This you've seen already. This is how it looks like after years of planning, tendering, and construction, and now we are in the end of the implementation phase and up and operating. This is top view. I will skip this. This is an important slide because, as you've seen, the installation developed out of the APPN, and there are many institutions working with many plant species and having different and many diverse research questions. We were forced actually to design the installation in a very flexible setup. So we can analyze there from multi-way plates via small pots, 250 milliliter pots, where we most of the time host Arabidopsis. But now we have trees there. I can show you in the room again. So, and up to 5 liter pots. And all this can be basically submitted to phenotyping in a fully automated way. So Easter, Christmas, and weekend. We have 2 watering and weighing stations. This is so important because this makes the installation super suitable for drought stress experiments because we can control in a highly random way, highly precisely the water levels of each individual pot. So we can set well-watered conditions and we can set different drought stress scenarios and keep the plants on a perfect set water level. So drought stress is a very hot topic for us. This is the environment control from 0 to 40 degrees, humidity 40-80, CO2 control from ambient up to 2000, and multispectral LED lights. Although we are operational only for 2 months, we managed to host 2 Austrian ministers of education and science. This is Mr. Fassmann, and Mr. Polaszek was 2 weeks ago visiting Phenoplant. This is my last slide. I have plenty of more slides, but this basically just shows you that we host most of the meaningful sensors, starting from RGB side, RGB top view imaging, physiological imaging like palm chlorophyll fluorescence and thermal imaging, and there's also a 3D triangulation scanner and 2 super fancy hyperspec cams that are operating in the FNIR and the sphere range, giving you in a super high resolution manner your spectral reflectance curves from 380 up to 1700. There are plenty of colorful slides more, but this I would like to discuss with you in the room. You simply go this direction, you pass the friendly porter, you grab an apple, and we are in E38. And there's also an in-house developed device by Sebastian, who is in the back of the audience. It's a Raspberry Pi controlled bench unit for crop plant phenotyping, site phenotyping with a turntable. Yeah, just drop by. Thank you very much.
Thank you very much. So, sorry, we had a bit of delay at the beginning, so we try to keep the schedule on. Just 1 sentence of concluding word. There's a lot of other companies we could not invite. The idea was not to make mega stuff, but I really want to thank all the partners that agreed to come and give you a good panel of what's possible in the phenotyping world, in the kind of low cost, very adapted to also the direct measurement that you do, which make a kind of good overview. And in my view, techno are just tools. What really matters are the humans that will interact with you to develop this tool on your own purpose. So that's the time for meeting, chatting, face-to-face meeting in all the rooms. So please go and spread out in these places.